In our last lecturette, we will discuss how we control cardiac output, which is the amount of work that is performed by the heart in response to the body's need for oxygen. Before we get into the mechanics of cardiac output, let's look at cardiac output in a big picture view, okay? So there are two ways to alter how much blood we are pumping throughout the body, okay? So we can either increase or decrease heart rate, and heart rate can be abbreviated for here on out as HR, capital. And then we can also alter something called stroke volume. And its abbreviation is big S, big V. And stroke volume is how much blood is squeezed out of the ventricle. Okay? And since cardiac output is determined by heart rate and stroke volume, multiplying these two factors together allows you to calculate how much blood is being delivered to the body at any given time. Now let's get into the details of stroke volume. So stroke volume is technically defined as the amount of blood pumped by one ventricle during contraction. So if you want to accurately measure how much blood is being pumped out of the heart, you need to calculate each ventricle separately. Now, stroke volume is calculated by taking the end diastolic volume and subtracting it from the end systolic volume, okay? And to refresh your memory, at the end of ventricular diastole, the ventricle is relaxed. And because it's relaxed, it is full of blood. Now, at the end of ventricular systole, we've squeezed our ventricles, okay? And so here the ventricle is contracted. And its contents now has been ejected into the great arteries. So all we're doing is taking the initial volume of blood and subtracting how much blood is left over after we squeeze the ventricle. And the reason that we need to subtract the second volume from the first volume is because under normal conditions, right? We're just walking around, sitting down, carrying on our daily educational business. The ventricles are only pumping about 60% of their volume. So there is still a lot of blood in the ventricle after systole, okay? Now, why does the heart pump only 60% of its ventricular blood to the body? Excellent question. I would pause the video and think about why an organism, which is big picture thinking in our field, why an organism would do this. So press pause now, think about it, and then press play when you're ready to hear the answer. And I already know majority of you folks are just ready to hear the answer. Now, the reason we don't pump all the blood out of the ventricles is because we use that blood as a blood reservoir, which means we save it. We save it for times when we have a sudden increased demand for nutrients like glucose and oxygen. Okay? Now think about it. If you pumped all your blood to the body, none of it is in the ventricles. Okay? It's all out in the great arteries and in systemically to the lungs and your brain and your appendages. And now all of a sudden you see a lion and you start running. your body would need more nutrients to run away. However, your heart would not be able to supply more blood that carry these nutrients because all of the blood is already out into the periphery. Okay? So we have a few blood reservoirs in the body, and the ventricles are one of them. And these reservoirs allow now our stroke volume to be changed based on demand. Now let's review how the autonomic system controls heart rate. Now, as a reminder, the autorhythmic pacemaker cells of the heart can generate electrical impulses all on their own. So we don't need innervation from the autonomic nervous system to have a heartbeat. And if some of you folks have dissected animals, you know that if the animal's alive, you put it to sleep and you pop out the heart will beat on its own, regardless if you've cut all the nerves and blood vessels from it. Now, remember, the autorhythmic cells fire at a specific pace that is determined by the pacemaker potential via the funny channels, okay? But if we have an increased or decreased energetic demand, we need to change the speed at which the autorhythmic cells are going to fire. And that's where the sympathetic and parasympathetic nervous system come in. And you know that the parasympathetic nervous system causes decreased contraction rate in the heart, and that the parasympathetic nervous system causes an increased contraction rate of the heart. Okay, so let's review the detailed mechanism of antagonistic control of the heart. Here we go, mechanism of antagonistic control of the heart. So the parasympathetic and sympathetic neurons are coming out of the medulla oblongata in an area called the cardiovascular control center, which we call the CVCC. Okay, CVCC. Okay, so here, when the second neuron of the parasympathetic system comes and innervates the heart, it's innervating the SA node. Okay, and so parasympathetic neuron, the second neuron, will release acetylcholine. And you folks know that acetylcholine, in this case, is going to bind to muscarinic receptors, specifically M sub H, okay, in the autorhythmic cell. And then that will cause the autorhythmic cells in the SA node to hyperpolarize. It's because I get a lot of potassium leaving the cell and less of the calcium coming in. And so you folks already know, this is all reviewed. This is nothing new. Hyperpolarization is going to cause you now to decrease the heart rate. Now, to increase the heart rate, of course, we're going to now turn on sympathetic neurons. And in doing so, you folks know that I'm going to start to release, for my second neuron, adrenaline compounds. Okay, so norepinephrine or epinephrine. And they're going to bind to the beta-1 receptors on the autorhythmic cells in the SA node. And of course, you folks know that depolarization will increase, and sodium and calcium are going to come in larger amounts, which allows me to increase the rate of depolarization and therefore increase the heart rate. Okay, so nothing is new here. This is all review. The only thing added is that we're now introducing you to the cardiovascular control center that's in the medulla oblongata, CVCC. In this slide, we will go over the mechanisms of how catecholamines, which are epinephrine and norepinephrine, increase cardiac contraction rate and force. Okay, so rate and force are two ways that the heart can distribute more blood. Okay, and if we need more blood pumped throughout the body, I can increase heart rate, which gives us more cardiac cycles per unit time. Okay, now remember, cardiac cycle is one systole and diastole. Okay, so one contraction, one relaxation. Now, for example, if my heart beats around 60 beats per minute, Okay, so this is 60 BPM. I can pump about 4 liters in a minute. Okay, but if I increase my heart rate to 100 beats per minute, I can pump about 6 liters in a minute. Don't memorize these numbers. They are to illustrate the point that increased heart rate leads to increased blood delivery. Okay, now contraction force is different than contraction rate. Okay, contraction rate is how fast I squeeze the ventricles, but contraction force is how strong I squeeze the ventricles. Okay, and the stronger I squeeze the ventricles, the more blood I can eject per ventricular contraction. Okay, so let's run some numbers. Okay, so let's say if I squeeze with a force of 5, and I squeeze with a force of 5, that allows me to eject 60 milliliters of blood. Now, let's say that I squeeze with a force of 8, okay? And that would allow me to eject 100 milliliters of blood every time I squeeze, okay? Now, the stronger I squeeze the ventricles, the more blood I can eject per ventricular contraction. And luckily, I have a reservoir in my ventricles that can be ejected if the ventricles contract stronger, okay? Rate versus force. As a review, epinephrine and norepinephrine will bind to the beta-1 receptors and activate cyclic AMP, which will lead to phosphorylation of calcium voltage-gated channels, which increase their open time, which allows more calcium to enter the cell. Now, as a side note, cardiac muscle has the ability to perform something called graded contractions. And all that means is in order to control force of contraction, cardiac muscle cells can individually contract a little bit or a lot, to have a little bit of force or a lot of force. And that is different than what we learned from skeletal muscle, where the muscle fiber either contracts or it doesn't. And the way that we now controlled force is by the number of skeletal fibers we recruited. Now, based on the information that cardiac muscle cells can do graded contractions, the more calcium I have in the cell, the more myosin will interact with actin and the more force I can generate. Okay? You don't need to memorize graded contractions and all the differences between cardiac and skeletal muscle. The take-home message here that is important for us now is the more calcium I have in the cell, the more force I can generate. Okay? So increased intracellular calcium will lead to increased contraction force. Now, activation of cyclic AMP also phosphorylates a protein called phospholamban, okay? And we are going to forevermore call it PLB. And what PLB does in its normal conformation is it slows down the activity of calcium ATP pumps on the sarcoplasmic reticulum, okay? So here, inhibits or slows, because these pumps are still working. They're just not working at their top optimum speed. So slows down sarcoplasmic reticulum, calcium, ATP pump activity. And if you remember, calcium ATP pumps are responsible for pumping calcium back into the sarcoplasmic reticulum, which allows muscles to relax. But if you slow the pumps down, it keeps more calcium in the cytosol. Okay? Keeps more calcium in cytosol. Okay? And if you keep calcium in the cytosol, it's going to prolong relaxation, okay, which leads to a slower heart rate. All right, now let's go back to our mind map here, okay? Now what's happening is when you phosphorylate PLB, okay, so let's draw that. PLB, and I phosphorylated it, okay? Now what's happened is you're doing something which I call, because this is an actual pattern that we'll see for the rest of the semester, is you're going to inhibit the inhibitor. Okay? And what that means is that PLB will change shape when it's phosphorylized. And now it can't perform its inhibitory function on the calcium ATP pumps. So now the pumps are very active. Okay? So I have increased calcium ATP pump activity. And if you increase the calcium ATP pump activity, they're going to be able to quickly sequester or suck up calcium back into storage. And the sequestering of calcium at a faster speed will accomplish two things. Number one, the faster I can suck up calcium, the more calcium I can store, which means the more calcium I can release in the next cardiac cycle. And the more calcium that binds to troponin means the more force I can generate. Okay? Number two, the faster I can suck up calcium, that allows me to remove calcium from the cytosol faster, which allows the cell to have a shorter duration of contraction, which means I can undergo relaxation and then another contraction at a faster rate, which leads to increased heart rate. Okay? Now, in the past, the concept of inhibiting the inhibitor has confused many students. So I would draw out your study sheet for this mechanism so you can practice and give your brain the opportunity to process this type of pattern of what results when you inhibit the inhibitor, which is a pattern that you will see again and again in other systems in your near future. Now, that's it for lecture 15. Make sure to fill out your muddiest point questions so that we can address any confusions in this lecture. Bye.